In this lesson, we are going to discuss lipids. At the end of this video lesson, you should be able to classify lipids as triglycerides, waxes, phospholipids, and steroids, and identify the uses of lipids in the processes of life. Oil, as we know, cannot be mixed with water. No matter what physical means we introduce to an oil-water mixture, the two will not mix. Fats from the food that we eat, just like in bacon, are notorious for being risky for our health, despite its appetizing taste and aroma. The oil and fats which we just saw are just examples of what we call lipids. Lipids are nonpolar biomolecules which mix poorly with water and are soluble by nonpolar solvents. Although lipids may have some polar bonds associated with oxygen, lipids consist mostly of hydrocarbon regions which are nonpolar. They mix poorly with water, which is a polar solvent. However, they are soluble in nonpolar solvents like benzene, ether, and acetone. Lipids are abundant in biological systems. They are classified into four subclasses: triglycerides, waxes, phospholipids, and steroids. Triglycerides are triesters of fatty acids with glycerol or 1,2,3-trihydroxypropane. To recall nomenclature on organic compounds, glycerol contains 3 carbon atoms which all have single bonds, making it an alkane, specifically propane. Each carbon atom has a hydroxyl group attachment, giving the name 1,2,3-trihydroxypropane. Glycerol will form large molecules of fats with 3 fatty acids. Fatty acids are carboxylic acids with the long aliphatic chains. A carboxylic acid is an organic acid that contains a carboxyl group with a side chain. In the synthesis of any biomolecule, a process called dehydration removes water molecules from the components to create a bond between the joining units. In this case, the hydrogen atoms of the hydroxyl groups of the glycerol component will be used with the hydroxyl groups of the fatty acids to form water. Remember that water contains 2 hydrogen atoms and 1 oxygen atom. Upon dehydration, a triester of fatty acids will be formed using glycerol and the 3 fatty acids. An ester is an organic compound in which a hydroxyl group of the carboxyl group is replaced by an oxygen. Thus, we have a triglyceride. And since we remove 3 water molecules, this will also be considered as products of the synthesis of triglycerides. The bonds between the glycerol and fatty acids are covalent bonds called ester linkages. Let's look at an example of the formation of a triglyceride with the 3 molecules of lauric acid, a fatty acid which contains 12 carbon atoms. To form a triglyceride, the hydroxyl molecules of the fatty acids will form water with the hydrogen atoms of the glycerol molecule. After dehydration, the triglyceride will look like this. Here we can see that on each carbon atom of the glycerol molecule has an attachment of lauric acid, making a triester of lauric acid. Now, the side chains or aliphatic chains of the fatty acids are classified into 2. The fatty acids can either be saturated or unsaturated. The major difference between the two is that saturated fatty acids do not have any double bonds on the aliphatic chain. On the other hand, unsaturated fatty acids contain a double bond which appears to be a bend on the aliphatic chain. Saturated fatty acids are solid at room temperature because the aliphatic chain are packed closely. Examples of these are lard or pork fat and butter. On the other hand, unsaturated fatty acids are liquid at room temperature because molecules cannot be packed closely due to the kinks. The double bonds of the unsaturated fatty acids contribute to the fluid behavior of the triglyceride. Examples of these are vegetable and coconut oils. Triglycerides may also be classified as either fats or oils based on the source. Fats are derived from animals like cows and pigs. Oils, on the other hand, are derived from plants like coconut and avocado. Triglycerides are mostly used as energy sources. Energy is stored in the bonds of the fatty acids. If a living organism consumes too much calories, this excess energy will be stored as fat tissues. If a living organism is deprived of energy from food, or if too much energy is used, for example in exercises, energy will be obtained from the stored fats in the body. Another subclass of lipids is wax. Compared to triglycerides, waxes usually contain only one fatty acid attached to a long chain of alcohol. The same process is followed to form waxes. A water molecule will be formed from the two components to form wax and one molecule of water. Here, the hydroxyl group from the fatty acid cleaves and bonds with the hydrogen atom of the alcohol group. Waxes are commonly used as coating to prevent drying. Cuticle is present in some plants, particularly in leaves, fruits, flowers, and non-woody stems to prevent excess evaporation. Plant cuticle also protects plants against UV radiation, chemical attack, mechanical injuries, and pathogen or pest infection. Beeswax is used by bees to form cells for honey storage and larval and pupal protection within the beehive. Candle and paraffin are some of the home applications of waxes. The third classification of lipids is phospholipids. These are composed of hydrophilic phosphate head and hydrophobic fatty acid tail. The hydrophilic part, or the water-loving part, is made of a polar molecule, a phosphate group, and a glycerol molecule. The hydrophobic part, or the water-fearing part, is made of one saturated fatty acid and one unsaturated fatty acid. Phospholipids are the major component of the cell membrane. The cell membrane is a lipid bilayer. Because of the aqueous environment in the cells, the phospholipid molecules orient themselves in a way that the hydrophilic head is oriented towards the aqueous part, and the hydrophobic heads are sandwiched by the heads since these are water-fearing. The phospholipid bilayer allows the selective permeability of the cell membrane to allow only selected molecules going in and out of the cell. Some molecules which cannot pass through the bilayer pass through the protein channels integrated within the bilayer. Lastly, steroids are lipids which do not contain any fatty acid. They are made of 4 rings, 3 of which are 6-ringed and 1 is 5-ringed. Steroids may have a negative connotation because these are used by athletes for extra strength during competitions. However, there are naturally occurring steroids in our body. A common steroid is cholesterol. This is used by the body to make vitamins and hormones. Not all cholesterol is bad. HDL or high-density lipoprotein is known as the good cholesterol because it helps remove other forms of cholesterol from your bloodstream. On the other hand, LDL or low-density lipoprotein is known as the bad cholesterol because a high LDL level leads to a buildup of cholesterol in your arteries. As mentioned, cholesterol is essential for the production of hormones such as testosterone, estrogen, and cortisol, all of which are considered steroids. Vitamins are compounds that are essential for the proper functioning of our metabolism. We are now done discussing the 4 subclasses of lipids and their functions. Let's now conclude this lesson by reviewing the following key points. Lipids are nonpolar biomolecules that dissolve in nonpolar solvents. Lipids can be classified as triglycerides, waxes, phospholipids, and steroids. And lastly, some of the functions of lipids include energy, coating, cellular semipermeability, and hormones and vitamins. And that ends our discussion on lipids.